All right. What will I tell you about today? Basically, I will tell you how we've contributed to speeding up drug discovery by improving the way protein crystals are detected. And in particular, here are the key takeaways that we will take away from this presentation. One thing is that we've improved the detection rate, so basically how precisely we detect those crystals. Second is a significant improvement in the computational resources needed for that. And the last point, which I personally find the most interesting, which is maybe also can translate to many of your problems that you might be facing, how to easily and efficiently fine-tune to new data. So basically, how to improve the generalization of the model. It's a recurring theme in many areas of machine learning, but in particular in healthcare applications, that you have a very nice model until you go to the next hospital, right? So we'll see how to deal with that here. So here's a more detailed plan of the talk, but let's jump forward and start with saying, who am I? So I actually work for Appsilon, and we're a scale-up company doing everything data. So we do data infrastructure, we do data visualization, we do data analysis, and in particular, machine learning. And in terms of technology, we have a large group specializing in visualizing data. In particular, we're very good at R-Shiny, and that makes us very good for working in healthcare because there is a lot of statistics involved in the models, and then those models need to be visualized. So Shiny is a very natural candidate for that. And then we also do machine learning, and that's the part we'll be focusing on today. And I happen to lead the machine learning department, where we mostly do computer vision, really. And then, as I've mentioned, we are focusing in terms of domain mostly on areas involving life sciences. You can read more regarding our case studies, for example, at the appsilon.bio page. So let's jump into the description of the problem we'll be tackling. And here, bear with me, I'm not an expert in crystallography, in protein crystallography, so I'll give you a layman introduction to that topic. If any of you knows more, please help me out, or tell me that I said something completely wrong in the questions section. But basically, the benefit is that because I have a layman view of that, it might be understandable for you as well. Yay, if you are not an expert. So basically, the idea is that in terms, when we want to discover new drugs, we want to make them very precise in their action. And a way of targeting the performance of the drug is basically to find proteins which can be used as in this process of delivering the drugs. So in particular, in order to find the right proteins, we need to prepare a lot of them. And this process is extremely complicated and time-consuming. And in particular, it involves a stage where we check which of the candidates actually crystallize. So which of the protein candidates, or candidates for being useful, do crystallize. And here's an example of an image with some crystals in it. I mean, you can clearly see there's some sort of crystallization happening here, right? Now, once we have proteins which crystallize, those actually can be analyzed, and we can understand their structure in detail, and then we can basically leverage those when designing new drugs. Now, the key thing to understand about this whole process is that it's extremely time and money intensive. So in particular, you can find quotes like several crystallographers can need sometimes up to multiple years, sometimes even given like 20, to actually find a protein crystal that is useful for the given purpose they need. And that means, in particular, that at this stage of screening whether your proteins do crystallize or not, every crystal counts, which is a direct segue to recall, right? You do need high recall as possible. Because if you're missing crystals at this stage, then your money went to, air. So you're wasting a lot of time and effort and money. So how can AI help? Well, you can imagine that what you need to do is identify crystals in such images, right? And sometimes they look nice, like here. You have pretty examples, right? These are nice crystals, and we can all say, oh, they are crystals. But of course, like in real life, crystals don't always look like crystals for human eye. Those are also crystals. It's just much, much harder to see them, right? So let's look by one. If we zoom in to that image, then if we zoom in even further, then we see this shape here. Oh, this is the crystal. Now, good luck identifying that if you have like hundreds or thousands of such images, and you actually need to do that fast, right? Now, if you look at the other one, you'll see another problem, which is that sometimes you have a lot of other structures which are actually not crystals, and then there is the promising crystal, which is worth a lot of your effort, and potentially money if you're a pharma company. But you do need to find that one somewhere close to the border. Now, in this one, you actually see a very different shape of crystals, which is yet another challenge to identify. Like it's all over the place, but the structure, like the visual structure that makes it a crystal, is actually very localized in some sense. So it's like everywhere, but then again, it's like the fact that the visual feature that makes it a crystal is very, very small. Yeah, and then there's all sorts of images which are taken from a different apparatus, for example, and then they look completely different, and the crystal is just some tiny deformation somewhere at the side of the image. All right, so what we see is that basically crystals come in many shapes, colors, scales. Sometimes they're blurry, sometimes there's low contrast, all sorts of very nice things that people doing computer vision love, right? So we defined what the crystals are. What is the rest? We need to identify crystals in what? So let's talk about the rest of typical data set. And apparently, apart from images with crystals, we have images which are, well, so-called clear. It's often done this way, right? So there are images which are labeled like this, and then they differ because, well, experts said there is nothing in them. But there are also precipitates, which I'm not sure exactly what that biochemically means, but basically there are some structures which are not crystals in there. And for me, many of them look identical to the crystal images. But, well, they do not for the experts, and they have very different meaning for the drug discovery process, and then hopefully they will look different for our models. And then there is also, well, the other category, my favorite. In every computer vision project I've worked on, the other is always the fun part. So basically there can be like some, artifacts, maybe some dust, maybe, well, you can find anything there. So there are also those images that you need to deal with. Your model needs to behave well when it sees crazy stuff. And now let's think of a case where we have basically a data set with thousands of such images, and we want to identify the crystals, right? So we can ask the foundational question, how good are humans at that? And this actually has been tested, and it turns out that people are not that good. And in particular, when we measure it, it's roughly 93% of agreement between experts whether this image presents a crystal or not. It sounds like, yeah, 90-something, but think about it. It's 7% of crystals just go unnoticed, right? Because well, people are not sure whether this is a crystal or not before they actually make further experiments. So this is a big problem. Now, there is a state-of-the-art model that is called Marco, that was introduced roughly 5 years ago, where a lot of large corporations and institutions collaborated to basically collect a huge data set. If I remember correctly, it's like several 100,000 images, and they are in those 4 classes. And then there is a state-of-the-art model which tries to identify which of the classes this is, so in particular, identify the crystals. And the paper actually claims a very nice result of roughly 93.5% accuracy. So you might think, well, it's great, right? I mean, humans agree on 93. We have something like that. I mean, it's a perfect model, right? It's as good as it can be. Well, not really. Because actually a known problem of this model is that it doesn't generalize. So even though you have already data from various sources, if you actually want to use it, you probably want to use it on yet another source. And if you actually measure it on yet another source, for example, this VIS data set, then the accuracy actually drops quite significantly. And these are example images from this VIS data set. But if you happen to work in a lab where the images look very different, then your accuracy can drop to very low numbers. And that's a real problem. You're missing half of the crystals. That means you have to do twice more work to get the results you want. And that's a problem. So, actually, if you look closely at the results of this model, you basically dissect them, you will realize that you can see signs of that already within this data. So actually this data comes from 5 sources, as I've said, And then if you look at the performance of this model on those individual subsets, it actually varies. And already on some of them, it's pretty bad. Right? So it's actually much worse than the average. And bear in mind that those data sets are imbalanced, right? They are not, well, if you average those numbers, you won't get exactly that. But what can we do about this? So, oh, yeah, one more thing, which is a problem, is that this model is very heavy to train. It takes, well, very heavy is, well, relative, but it's the remaining time? Yeah, it's the remaining time, good. Right, so it basically took 40 GPU days to train this model, which means it's quite hard to, well, for example, maybe adjust this model for new data or something. So what we've done is first we've basically redid the job in the same setup. So basically we wanted to answer the question, can we do better? Right, so can we take this data set and just beat the state of the art? And yes, we can. Before we go into the results, the few key improvements that we've made that I think contributed the most is one, we've used the transformer model and not the inception-based model that was used in the original result. We've used the pre-trained model instead of training from scratch because, well, that's what you should do in computer vision. We also adjusted basically what the model should look at to the case because the model is actually not judged by its accuracy. It shouldn't be judged. It should be judged by its recall. And basically the result is that, first of all, within 1 day instead of 40, so in a much, much shorter time scale, we got a model that has matching precision, so it produces as many false positives as the state of the art, but then it has an improved recall. And you might think of like, that's not such a big change, right? It's like close to 90. Well, but if you look at it from the perspective of the actual end user of this model, then this means that the number of crystals they will miss is lower by 30%. So roughly 1/3 now is going to be discovered. 1/3 of what was lost now is going to be discovered. So this is actually a big improvement. All right, and as I've mentioned, we've shortened the computational time quite a lot. Okay, so that is all fine, but it's still not really a very much realistic scenario, right? In a realistic scenario, it's not that you have a data set on which you want to measure your performance. You actually want to use a model in your own lab, right? Or, for example, you're working on a project for someone and they want to use your model on their data. And the question is, what happens if the model is actually meh, like we've seen the state of the art is on some type of new data? On some it's decent, and some it's quite poor. And the thing is that you can actually leverage something that has been used or has been developed for large language models, and that is LoRa. So I'm not sure if you're aware of that, but LoRa is basically low-rank approximation, right? So we basically, I'll tell you a bit more about how it works, but basically it's a thing which allows to fine-tune transformers efficiently. And it's been mostly used for language models, but it can also be used for vision transformers. And that's what we've done. And what it allows us to do in a moment, but basically to be a bit more technical, what it does is that if you think of your model as, like internally, if you look at a part of the model, and there is some weight matrix, right, which contains the weights of the pre-trained model, and it potentially took you, well, half of the internet to train. Then if you want to fine-tune your model to a specific task, you can actually, instead of doing it the classical way, so adding more data and fine-tuning the entire thing, you can add a small component, which is small in the sense that you are actually replacing this original matrix, which is D by D, where D is supposedly rather large, by a matrix that is a multiplication of two matrices. And then each of them is actually not square, but rectangular in such a way that you're reducing basically the number of parameters that you're training. And then you're fine-tuning only this small addition. This has the benefit of basically a huge computational shift or saving. That's one thing. And the other, it actually helps a lot with so-called catastrophic forgetting. So basically you don't touch those weights here. So, well, waving hands, you can say that your model probably will remember what it already has learned, and then still is able to get some new knowledge in this part, which basically is hand-wavy, I agree. But, well, if you check, that's how it works. So, Yeah, so with that, you can basically shorten the time of training or fine-tuning really large models to manageable time and data. So the important thing is that because you have a lower number of parameters, it doesn't only shorten the time you need, but it also can drastically, as you'll see, shorten or make smaller the amount of data you need for that. So in particular, let's go back to those two data sets on which Marco was pretty bad. The first one on which it was, decent is the Viz data set. And basically, if you look at the accuracy, I think, is given here. Yeah, the Marco benchmark is like 91% on this data, right? But basically, with our LoRa fine-tuning of our models, it's enough to take 60 images from this data set to basically already get to the level roughly of 92, 93%. I think it's actually 93. It's an old graph that I showed here. So basically, we're already at the, human performance level, so to speak. Those are not directly comparable numbers, but you can think of it this way, or at least the declared performance of Marco, just with 60 images. And that's something that any lab which asks you to do or wants to do already has, because they typically, they know that the model doesn't work, right? So they had to have at least some data. So just with a very tiny number, you can already boost the accuracy. And please note that with 2,000, which is not unrealistic, you can already get to performance which is way higher on this particular data set. And then even more remarkable difference is on this harder data set, because it's kind of like, you can think of it as being further from the distribution of your training data. And then adding 60 gives you a boost of like 10 percentage points, which is huge, because remember, we're talking about crystals which are extremely precious for the drug discovery process. So basically, what that means is that, first of all, we've shown how to reduce the number of missed crystals using much less computational power. So in case you actually do have a lot of data, you can do those things and then have good models much faster. But more importantly, I would say, there is a way of going beyond your distribution of your training data, right? You can leverage this method, and we have done this also in other projects, and it does wonders. You can actually, because this is a realistic scenario, quite often you do have some labeled data, or you can acquire some labeled data. It's relatively cheap to get some. It's very expensive to get a lot of it. But if you get some, of course you need some for testing, and then just about the same for training, and this is enough to already get quite a good result. So we're happy to deploy this. We're now in discussions with partners to basically use this on real data. So because here it's still on a data set we got from the literature, but we'll be testing this, and probably you'll hear more about this when we test it on actual data that is needed by people. And here you can scan the QR code and read more. If you're interested, you can reach out to me directly. Thank you for your attention, and I'm happy to take questions.
Thank you so much, Jędrzej. Do we have any questions from the room?
Hey, hello. Thanks for the talk. My question is about actually how common the crystals are, because the accuracy is rather big, but if the crystals are very uncommon, then it should be big. But
no, they are not. So the thing is, there are two angles to this question. One is how common are they in the data set, and second is how common are they in real life. So in this data set, it actually depends. So if you remember the Marco data set, so the main one consists of 5 subsets, and the distributions. This is actually a 4-class data set, so crystals is only one of them, but the distributions vary quite a lot between them. In all of them, it's very unbalanced towards the crystals. So there's just roughly speaking, 10%. If I have to give an estimation, that's roughly 10% of crystals. But in some, it's actually even less, if I remember correctly. And then in this 2 extra data sets, so the Viz and UV, those data sets, if I remember correctly, it's actually balanced there, I think. So those data sets are quite small. It's roughly 3,000 crystals and 3,000 non-crystals, if I remember. Well, maybe the data set itself is more, but we downsampled the other classes because we only need roughly the same, right?
All right. And in the real life? Yep.
in real life, I'm not an expert. And to my understanding, this really depends on the exact setup. So what type of proteins you're looking for, what is your procedure, how hard it is to synthesize them, how good your team is, because this is wet lab, right? You actually do need to do that with your hands, or set up the machine right, calibrate it. So I'm not sure about this, but I think it's even lower. So it's closer to like those numbers here. So those accuracies are not that bad, actually. I mean, some of the images are easy, right? Just some of them are very, very hard.
Okay, so I wanted to ask whether you had a chance to test LoRa versus training head of the architecture only and freezing the weights of the backbone. So this is the first question. And the second one, what is in reality LoRa architecture? Because on one side we have the freezed parameters of very extensive transformer architecture. And what is LoRa? Is it just simple multi-layer perceptron or something more?
No. So, okay, there are two questions, right? One is LoRa versus fine-tuning. So the thing is that this data, like, you can do fine-tuning in different ways, right? But I've never seen an example of fine-tuning, like actual fine-tuning of all the weights or like maybe some frozen layers with like 60 examples in a setting which is so diverse. So, I don't think that there's any chance. I'm not sure we run experiments on like 60. We've run experiments on the entire data set. So have everything we have, and let's compare it to fine-tuning. We've fine-tuned transformers, several of them actually, I think SWIN and VIT. And we've fine-tuned convnexts, a few versions of convnexts. The convnet for the 2020. Actually, it's a V2, but never mind. So yes, you can compare that, and LoRa beats it by far, especially in the low data regime, like low data size regime. So that's one question, and the other was LoRa. What is LoRa? How it works? Right. So basically, you can think of it the following way. So imagine the original architecture, right? It has some weights in it, right? You can look at individual layers as sort of matrices, and then what you're doing is you're kind of next to it, imagine another matrix, which happens to be a product of two, so it's actually fewer numbers, right? Because the extreme is you take just two vectors, right? And you multiply them to get the matrix, right? If you have two vectors, then you have okay, let me do it this way. Instead of drawing in the air, let me point. So if you have a matrix of D by D, and you take it to the extreme, you are adding a vector of D and a vector of D, and you just multiply them like the matrix-wise, and then you get a matrix of D by D, right? And you can add those two, and then freeze those, so the original weights, and just train 2D parameters, basically. So instead of D by D, when D is large, 2D is much smaller than D by D, by a factor of D half. But yeah, so that's it. And of course, in real life examples, I haven't seen people doing like vector times vector. They do like a rectangular matrix by a rectangular matrix, but still. Yeah, and you can add those LoRa matrices, LoRa adapters, they are sometimes called, at different layers. You can add it all, you can add it some. Yeah, it's a kind of hyperparameter decision of sorts.
Thank you. Any more questions?
Just maybe one comment. In some sense, it's sort of like training on residuals only. Like it's different than the residual networks because there is no skip connections. But in some sense, you're kind of like extracting the pre-trained base of those numbers and just training for the like tiny modifications. Because also you're initializing like close to zero, right?
Next question. So I would like to ask you one thing. Can you comment a little bit on, not in the technical direction, but more on the business direction? So can you comment on any results of your work that are actually practical? So do you have some numbers to put on it? So your customers increased the tempo of providing new medicines or lowered the cost or something like that?
Yeah. So in many projects from my team, yes. In this one, no, because we've actually done this research in the past 30 days. And we've just given, this is the second time we've talked about it 2 weeks ago at Harvard, and now we're talking here. And we are literally in the process of going to, arranging setups with research labs and commercial labs to basically get those numbers, right? To actually, help them, or, help them research, help them make money. So I'll get back next year with results.
Do you have any expectations? So seeing these numbers that you increased on the level of accuracy and so on?
Yeah, what I can tell you is that typically when you look at like the cost of synthesizing a protein of different type, like there are bacterial proteins, there are I think animal proteins and human proteins that are kind of typically listed there. And then also human proteins are very different types. There are those membrane proteins and then different types. I'm not an expert again, but those costs of those are in hundreds of thousands of dollars. And for membrane proteins, which are actually the best candidates for drugs, as far as I understand, this goes into millions of dollars. So, not missing 30% of the ones you've missed can be really, really saving a lot of money. And on one hand, it's a lot of money for the pharma company, but on the other hand, it's bringing drugs sooner. And we've seen recently with the pandemic that sometimes time counts. And, it matters that we get the drugs earlier.
Okay, thank you very much once more.